Okay, I apologize if that
Complementary RNA
got a little
strand called
glitchy.
messenger RNA.
Sorry, I apologize if that got a little glitchy. My battery ran dry on my laptop. So let's take a look at this again. We're comparing DNA to RNA, and we're comparing the single strand of RNA to the double strand of DNA, and we're looking at a process called transcription here, where the RNA is actually serving as like the pink slip from Miss Hager to copy the message down, to copy the main message down, to take it out of the nucleus into the cytoplasm. Remember, RNA is like a pink slip. It's a copy of one of the sides of the DNA. Again, this is like Miss Hager copying her thoughts or her master plan onto a pink slip. So let's watch this again.
Has to complement the DNA. More about that in our protein synthesis video. So if I still had the original portion of DNA here, what would be the complementary RNA bases? The answer, so remember, it's asking for RNA. No thymine here, it's uracil instead. Apples under helps you remember A with U. Car in the garage helps you remember C with G. So in this portion, reading in the direction shown on this image, we'd have these RNA bases.
Okay, again, remember the difference between RNA and DNA. DNA is double-stranded, RNA is single-stranded, but RNA serves only as a helper molecule to copy the message of DNA to get it out of the nucleus. One of the main differences of RNA is the fact that it has uracil instead of thymine. So instead of this apples and trees business, if this is my DNA down here, I can have apples, I can have trees and they say garage and cars. I say cows and grass, or grass and cows. If this is my DNA, if this was another side of DNA up here, I would have T's right here, and I would still have A's and C's and G's here. But because this is RNA up here, remember, I'm making the pink slip. I'm copying the message down to get it out of the nucleus. The substitute chemical in RNA is called uracil. So uracil substitutes for thymine. So instead of apples and trees, I like to say Alabama University. I know the video says apples are under the tree. Whatever you want to remember this by. The fact is, U substitutes for T when we're talking about RNA. So down here we have a DNA strand. Up here we have an RNA strand. Same thing is happening down here. Here is my DNA. These would be my A's and T's and C's and G's and along the line. This up here is my messenger RNA. This is the pink slip being made. This is Miss Hager copying her code to get it out of the nucleus.
Notice the answer is similar to the last answer, but there are uracil bases here instead of thymine. One last thing. Our models here, they're unable to show the beautiful 3D shape, the exact numbers of bases per turn, the chirality, but we've left you some links to explore because the goal of our videos is always to introduce the topic so that you can discover all the fascinating details and exceptions. Check out our further reading description to keep that curiosity going.
Okay, and some of these are embedded in my PowerPoint anyways. Let's have a look back at the PowerPoint. We're talking about this process. So the whole thing we've been talking about today in this part 3 video of the PowerPoint for chapters 12 and 13 is known as transcription. Again, this is like Miss Hager writing the pink slip. We have to get the DNA message out of the nucleus and into the cytoplasm, where we can actually start building a protein. All right, so this is the process of transcription, taking DNA and copying the message into RNA. The differences again are uracil is now substituted for thymine when we're talking about RNA compared to DNA. Let's take a quick look at your chapter. This is your chapter 13.1 from your textbook. And again, make sure you're going over your textbook. Good way to kind of think about this chapter 13 business, this business of transcription and translation, which we'll get into later, is think about the master plan for your cells. You don't want to ruin the master plan. You want to keep the master plan safe and sound in the nucleus, but you need to make copies to get the copies out to the workers to build whatever you need to build. So this is like Ms. Hager with the master plan, staying protected in the nucleus, staying protected in the office, but still keeping a copy or making a copy to get out to the rest of the people that need to do the actual job. So This would be the process of transcription, taking the DNA's master plan and copying it onto the messenger RNA's copy of the plan. Notice the plan still stays safe in the nucleus. Also, down here, and this is your chapter 13 stuff, please make sure you're reading your book. Another way to look at this, same kind of deal. This is our nucleus. Remember, this is the inside or the inner portion of the cell that contains the DNA in eukaryotes, cells that have a nucleus. Here's my DNA, my double-stranded DNA. This is like my Miss Hager with the master plan, but I need to get the master plan out of the cell, so I'm going to make a copy of this stuff called RNA, in this case, messenger RNA. There's an enzyme that helps the whole process called DNA polymerase. I don't know if you can see my cursor here. This enzyme helps this copying process take place, but all we're doing here is making the message available to go outside of the nucleus. The differences as far as DNA and RNA, look at, again, DNA double-stranded, RNA single-stranded. DNA has thymine, RNA has uracil down here. And again, I don't know if you can see my cursor, but uracil is right here. So uracil is a substitute chemical for thymine when we're talking about RNA. DNA has no uracil, RNA has no thymine. It's a substitute chemical. Think of it as like a little difference in the lettering, okay? Lastly, or almost lastly, when we make our pink slip, it may need to be edited. When we make our messenger RNA, we may need to edit it, just like you would edit a paper that you write for English class. You might take some things out, you might add a couple things and make it more perfect. So think of a rough draft versus a final draft. And the same thing takes place when we make messenger RNA to make sure that that message is perfect and that we build the correct protein based on the correct instructions. All right, going back to our PowerPoint here, we're almost done, guys. Again, this whole process was known as transcription, and just an overview of what we just discussed. This entire PowerPoint part 3 was about RNA, different forms of RNA, and the way it's different from DNA. However, it has many similarities as well. It's a helper molecule to get DNA out of the nucleus, or DNA's message or code out of the nucleus. The ultimate goal, the whole thing we're trying to do here, is build this protein. I'm going to go back to our analogy from earlier. If you remember, our top 10 group was all put together by Miss Hager's master plan that came from the nucleus, that came from her office, that went out from a pink slip and from office pages to get these kids in the right spots to make our photograph, which was our ultimate product. So again, our analogy. The DNA is the ultimate master plan that sits inside the nucleus. It copies its code to messenger RNA. Remember, there's some differences there because RNA is only single-stranded and it only has uracil. It does not have thymine, but still it has adenine, guanine, and cytosine. Remember, it has A and G and C. It's just that it substitutes uracil for thymine. That messenger RNA is then kind of passed off to a chemical called transfer RNA that goes and gets the individual components that make up a protein. That protein is then built in the correct sequence or the correct order, or the correct shape, by the way. It gives me the correct chemical reaction that gives me the trait. I know that's a lot to chew on right now, but go back to the beginning of this PowerPoint and then also stay tuned for part 4. That will make more sense. An easier way to say this is Miss Hager has to write the pink slip, gets her message out, gives it to an office runner that takes the message and goes and gets the individual students. Notice these are going corresponding one-to-one here. The students then are assembled in the correct order. That's very important that they're in the right order so that they have the right shape. Finally, those students, the task for them is to pose for a picture, and the final product is to get that picture. So looking at this analogy, the DNA is similar to Miss Hager. The messenger RNA is similar to the pink slip. The transfer RNA is the people that do the actual plucking of the students out of each classroom. The amino acids, of course, are the individual students. The protein is that team of students or that entire group that's assembled in the correct order. The chemical reaction is the thing that the students are going to do because they're properly placed in the right order to perform their task. And the final trait is the actual product that we produced. In this case, the picture of the top 10 to put into the yearbook. Okay, I hope that helps. Again, as always, anything you need, let us know. Contact your biology teachers. We're here for you. Get in any missing assignments. Stay well, stay safe, and stay sane as well. All right. Have a great day, everybody.